So good morning. So we'll continue with the lecture, that is valorization of solid side stream, and we'll have a bit of recap. Yeah. So first, for recap for yesterday's lectures, we did discuss about what is waste. Yeah. How we can reduce it or reform or transform. Yeah. So these are two important elements. Still, before we propose to reduce the waste or to transform the waste, we need to know how much waste is generated and what is the quantity of waste is produced. Then only we can draw down the strategy, and that's the way the policies are made. Yeah. So quick recap for food loss and waste. First thing what you need, this is the crux, whatever we discussed yesterday. This is one is action planning, identify the problems. Yeah. What are the problems? Set objective and KPI. KPI stands for key performance indicators. That means where all you can improve the process. Yeah. Then develop action plan, that is define how you can reduce it. For example, freezing and thawing, how you can reduce it, or you can eliminate the freezing and thawing process to reduce the spoilage. Then you again target how much is produced and how much is wasted and how we can compare with whatever baseline. So that because all the policies are made for the future. Yeah, that's one. Then whatever action plan you have made, you implement those actions. It's pretty much straightforward. Monitor the KPI and to see how far you are from your target. It's very similar when you have to prepare for an exam. What all you have to study, how many lectures you will study, what day, and then you implement that plan, and then see how you can further improve that particular action plan. So that's always a live process, and then analyze all the factors which are limiting for achieving those targets. That is, amount of food redistributed or reused, number of food insecure individual, per capita waste generated, or food waste generated per number of meals served. So if you'll go in a canteen and restaurant, it says there's a figure, it says how much of the food is wasted. Yeah. So we have to clean our house first before solving the global problems. Correct? So first you have to see how you reduce your own wastage. In your house, meals are cooked. You ask for 4 idlis, you eat 2 idlis, nalla illa gone to bin. Yeah, right? So if you are correct, then only you can make other people's correct. Yeah. And then generated per kg of product and how you can reduce the wastage. And then again, how much it is a cost, environment benefit, ecological benefit. We have done all those things in different lectures. Yeah. So this is very 3-1. First, action planning, what you should do and where all you can implement the technology, that is application of technology. Second, how you are going to run it through. And third, once it is achieved, to see how effective and how efficient you are. Yeah. You can put anything into this plan. Suppose this is your roadmap for your exams or writing a research paper or for anything. First you set, okay, every week I will study 2 hours every day. Yeah. Your friend calls you for a movie, gone 4 hours. Yeah. So then you improve the plan, you change your plan. Yeah. Okay, let me study in night, not in the morning. Yeah. So like that you make a changes, and then you see how efficient you are, how effective you are, and then you develop your what are the barriers or what you need to allocate additional resources to complete that particular action. Yeah. Is it clear in that? This is nothing rocket science. It's just arranging things to make it in a systematic order. I'll give you a very simple example. If you'll see, if there is any flood or tsunami or anything happens, you have seen that always army is called in. Yeah. Why it is so? Do you think army is more efficient compared to civilian police? It's not true. The reason is there is always a process called SOP, Do you know what is SOP stands for? Very good. It's a standard operating procedure. So any work you do, if you create a standing operating procedure, revisit it and improve that standing operating procedure, then you don't have to deviate it. It's not that you have to go in a lab to do experiment. That is called, when you start Havan or Yag, and then that time you're just missing something. Yeah. So you arrange all the resources, then you plan it, you do a virtual run and then execute it. Take a simple example, you would have attended any of those wedding ceremonies. Yeah? How long does it take to plan a wedding? 6 months, 7 months? Yeah? Execution takes 3-4 hours. Yeah? If planning is not correct, execution will have a problem and there is a point of no return. Yeah? Same thing happens for every work. Yeah? So planning is very, very important. You can take longer to plan. Execution will be very smooth if planning is proper. Yeah? And be prepared for worst case scenario, right? So anything can happen. For instance, we are supposed to start 9:30. There was some repair work going on. There was no cut. So we should have action plan ready. That is plan A, plan B. I'm not talking about the movie plan A, plan B. Yeah. So that's very critical for doing so. Or any experiment, any research work, if you follow the systematic order, something goes wrong, your result is not correct, you can always go back. Yeah. Otherwise, you'll say, oh, I think I added 10 grams. Oh, I think I soaked it here, right? So any deviation if you're taking, if you're making a record, then it becomes easier. So that's why document management plan, DMP, is one of the most important aspect of any research work or any work we carry out. Yeah. And that works based on this principle, FAIR. Yeah. I'm mentioning this, but this will come back again in lecture number 20 or 22. What FAIR stands for? Whatever notes you are making, yeah, you should be able to find it. Findability, yeah, correct? You are able to find it. A means accessible, yeah. You stored it in a laptop, but you can't access it, yeah. Second important principle. Third, interoperability, that is interpretation. That means whatever you're making short codes and other thing, you should be able to interpret it what you have written. Yeah. Banking sector works in a similar principle. If one accountant is not there, that does not mean you don't know how much money is there in account. Yeah. So that is carried forward whosoever takes the desk. Yeah. So you should be able to interpret that. R means what? Reusability. Yeah. That means should be able to reuse it. Yeah. So data management plan is for FAIR principle. Yeah. Findability, accessibility, interoperability and reusability. Yeah. So this is, just remember that this is one of the most important principle for any data. And if you suggest any other improvement, that can be done. Yeah. Now coming back to actual system. Yesterday I asked a question, what is the difference between protein concentrate, isolate and hydrosolate? Where are the dairy expert or the person who is doing the protein extraction? You, yeah?
Sir, protein isolates are protein made by removing some of the carbohydrate and fats and some non-protein components. It contain about 90% protein. Protein concentrates are the proteins are produced by removing some of the non-protein carbohydrate, but it contains major amount of fats and carbohydrate. It contains about 60 to 80% protein. And protein hydroxylates are the protein which are made by breaking down some proteins into smaller chain of amino acid by process of hydrolysis.
Okay, and this is, and what will be the protein content of protein hydrosolate?
They are about 80 to 90%.
For hydrosolate? For protein hydrosolate? Okay, 80 to 90. How many of you agree with your colleague? He is correct, but yeah, any other addition? Yes.
Sir, hydrosolate can be 50 to 85, 50% to 85.
Now this is only the target. The question is how you make it, yeah? Protein concentrate or protein rich flour and all those elements, take any biomass, yeah? So if you take, your friend is extracting protein from chickpea flour, yeah? So protein content of chickpea flour is? Okay, I'll put it 22%, right? Yeah? If we remove a carbohydrate, as your colleague mentioned, yeah, 22% this, 60% say for example your carbohydrate, 3 to 4% fat and similar 3 to 4% ash, right? If you remove a carbohydrate, this 22% will increase to 50 to 60%, yeah? That becomes protein concentrate, yeah? If you further purify it, it becomes protein isolate, which is always above 90%, that is correct there, yeah? Then if you use an enzyme to break the protein, it becomes protein hydrosolate, yeah? So percentage can go almost up to 99% also. Sometimes protein hydrosolates are prepared from protein concentrates or from protein isolates, yeah? Now when it comes to the definition of this, there is no clear-cut borderline, yeah? So what we call it protein rich food. Technically anything which contains more than 40% protein will be considered as protein rich food, yeah? Yeah? On what basis? Fish never contains more than 20% protein, but it's a protein rich food. Meat is not having more than 20% protein, but it is not as a protein, but sorry, it's still called as a protein rich food, yeah? So you have to be sure what definitions are. But in the case of dairy, definitions is very clear. What is whey protein isolate, whey protein concentrate and whey protein hydrosolate, yeah? So these definitions are not the definitions written on the wall that cannot be changed. Does it make sense? Yeah? So some of the protein extraction experiment you will be carrying out on a fish in next 1 or 2 days, even from shells, yeah? So that was the question related to this. And we will cover all these preparation methods and all in coming lectures. How do we isolate protein, how do we concentrate proteins and other elements, yeah? So what is content of today's program? One, what are the solid side stream and what are valorization strategies for solid side stream? So yesterday when we were covering about valorization strategies, about solid stream from different aspects. One was your processing stream, consumer or household is a similar waste, as well as the production side of it, yeah? Now if you take aquaculture, this is a simple aquaculture system, a representative of aquaculture system, whether it's a pond system or IMTA system or as what you described the other day. What is the assumption moving forward? You saw that statistics, aquaculture is increasing day by day, or every year aquaculture consumption is increasing. So there is a more demand for sustainable seafoods. Consumers are willing to pay more, especially when it comes to European consumer, which is produced sustainably. Preference is given more for locally grown food compared to supermarket or bigger brands. A. B. Again, the preference is given to the local growers, local processors, rather than buying in a centralized chain. We are going back slowly to the older one, because earlier you used to have a small shop near your house. You go and make a list and get all the ration and everything, whatever you need for your daily activity. Nowadays you go in a supermarket with a big trolley, pick whatever you want and you come back, yeah? Now slowly things are going back to the older story, where we has to have like record is there, 1 kg rice, 2 kg this, 100 grams of rice. This is the way it was older time shopping was done, yeah? Availability of sustainably produced biomass, yeah? Lack of innovation in aquaculture wastewater, yeah? So these are some of the assumptions. Then we are looking for valorization. And then the problem, currently there is a aquaculture wastewater as well as aquaculture solid. Yesterday when we were talking about aquaculture, we saw what are the waste we generate in aquaculture production system. Feces, uneaten food, yeah. So if you remember those strategies when we are talking about lecture number 8, yeah, valorization for aquaculture, processing and consumer end, yeah. So you have to just remember what was discussed other day, because there will be all continuation, yeah. So it is a base and then we build a block on it. If you remove any middle block, building will collapse, yeah. So that is very important to have a full cascading approach. Second one is alternative food and feed based ingredients, that is novel sources of biomass. So what we propose for valorization, one is valorization route for aquaculture wastewater, land-based macroalgae production, yeah, because macroalgae production needs a sea. Well, what about landlocked cities or landlocked countries? How they can produce macroalgae? So we should have a provision to produce it on land as well, yeah. Complete biorefinery of macroalgae, new food and food products, new feed formulation, zero waste model for macroalgae, seafood and other streams, yeah. So this is what we are going to cover in next subsequent lectures along with the technology. So what it will help in achieving sustainability aspect of current aquaculture practices. So it will provide you new improve sustainability current aquaculture practices, new ways of cultivating macroalgae, yeah. What we'll get? New food and feed, and help environment some way or the other, and ensuring circularity and waste mitigation or minimization, yeah. So this is what aquaculture stands for, right? Now going back to side stream, we did discuss about this image other day. So what are the solid stream we get from fish? You can read it. Skin, head, tail, what else? Frame, stroke, bones. You can say loudly anyway, because otherwise he'll be carrying the mic everywhere, yeah. Bones, this is from fish. And whenever such questions are asked, what are the side stream produced, yeah, straight away think of the biomass and see what portion is eaten, what is not eaten, yeah, rather than doing the guesses, then you miss some of the point, yeah. So everywhere if you follow a system, it makes things easier. Either you memorize it or you need to know this. So there is always an argument, I did not get a good marks because I can't memorize it, yeah. But that's not really a valid argument, yeah. You do memorize who has taken money from you, yeah, and we tend to forget who has given money to you, yeah, not money or any stuff. So you can memorize, you have to understand, then you have to memorize. If medicine doctor say, oh, I can't memorize what is a medicine, what is a dose, I have to give it to you, but I understand what medicine is required. Does it make sense? Yeah. So that's not really a valid argument. If you are really interested in, you will not forget, yeah. If your only interest is just to clearing the exam or clearing the course, then you won't remember, yeah. So many times we'll watch a movie and we'll never forget the dialogue, yeah, lifelong. It's just because our focus is on that one. Brain is having enough of capacity to retain everything, yeah. So in case of fish, head, tail, skin, visceral organ, plus your frames and bones. In shells, shellfish, outer shell, attached proteins, all those things will be side stream. In seaweed and duckweed, microalgae, will come back to that one. And there also waste is whatever is not consumed. If you consume whole of the carrot, there is no waste, yeah. Whereas if you peel a carrot to make a carrot halwa, then peel is a waste, correct? Apple pomace is only waste if you are extracting apple juice. If you're consuming whole apple, then only the seed is the waste. Yeah. So all the waste depends upon processing strategy. Yeah. So there is not the thing that there will be some waste, because some places or certain part of the country, I don't remember exactly, where the whole fish is fed to some people as a disease or treatment to asthma or something. I've seen. Sorry. Correct. So you just fish is fed it directly.
Specific period when there is a transition of sun in the month of June, means the Sankranti to another era. At that particular day, in that particular time, that fish is given.
Okay.
Means there are lakhs of people in Hyderabad.
Okay.
Yes.
So see, this is I heard about it, and that means that's a whole live fish. Yeah. And in some cases, a whole live fish is consumed. That means you are eating its viscera and everything, right? Whatever is inside, the bones and everything is consumed, correct? So that's a whole fish. So there is no waste, correct? But if you are even like a big whale, if it consumes large amount of fish, they do not segregate the stuff, right? They eat everything. Am I correct in saying that? So depending upon what all can be digested based on the waste. So waste is generated based on processing strategy. Yeah. It is not that there will be always a waste. There can be waste, or you can avoid the waste. That's the reduction and this one. But that reduction should not be that, oh, we should not waste it, let's eat everything. Yeah. Then you end up in another problem. So you have all these waste, and then it generates your liquid side stream, which we'll cover afterwards, and we'll go through all these extraction technique and what are different value added products can be obtained out of it. Yeah. Now look at this particular diagram back again to the fish. Muscles 15 to 20%, skin, fins and tails, trimmings, viscera, bones, heads. Yeah. So when we are talking in this particular lecture is all about solid side stream plus the biomass. Yeah. This is using better what we already use. This is unlocking the potential of seas and ocean. Yeah. That is what the solid stream, what we are going to cover now, and after that it will continue as liquid food. So we'll just continue with the sequence of lecture. Yeah. So I'll come back to this one again, but do remember what are the wastewater we get out of it. Yeah. Filleting water, stick water, blood, brine, that is if you are using a brine solution, cooking water. If you cook it, if you boil something, there is always a water. If you cook a rice, you separate sometimes rice water. What it contains? Starch. What do you use it for in a house? Putting your clothes into it. Yeah. I do remember that used to be what you call that dupatta, like school dress. Yeah. That used to go into that so that it's having a perfect crease. Yeah. Right. What do you call that process? Textile. Starching of the textile. What process is called? I know in Hindi it's mard or something is added in. Mard, that's basically coming out of the rice, that's a starch. Yeah. And even it is used also in, you may not be aware of it, but still all those colleges which goes for government inter colleges and all, I do remember the blue color dress and having a specific uniforms and dupatta. Even my sister used to go there, then they used to do that. So starch is extensively used in textiles. The starch is also used extensively in the paint industry. Yeah. So that's one, that is what sometimes you get a cooking water with the protein and other polysaccharides coming into it, or the water which is coming during the drying, that is evaporation losses. So these are some of the liquid side streams. Now, if you remember, we did cover this briefly. Yeah. So if you have a fish, whether it's wild harvest or aquaculture, it goes to the industry. Yeah. What we do, we make fish fillet. Yeah. And this fish fillet is produced and which is consumed. So the losses will be visceral organ, heads, tail, skin. Yeah. Plus the water is one thing. Other one is a fish cake. That means if the fish is minced, you mince a fish, you mix it with your wheat flour or different ingredient and you make a cake, yeah, and that's called fish cake. Or the same thing like what aloo tikki is made, similar type, potato is also added into the fish to make a fish cake. They're different ingredients, that's all culinary sciences, yeah. Then you have sausages. That means the same minced fish can be stuffed into a casing. That casing is either made up of intestine lining or is made up of cellulose or made up of different material. You stuff it, you steam it or you ferment it, that's called sausages. Or the fish, you make a sauce of it, yeah. And then, or else ready to eat product, fish chips and all those things, yeah. Can any of the fish eater can suggest one or two more products coming out of fish? Am I missing any of the product here? To avoid that question, normally I write ready to eat. Everything can be covered into ready to eat, yeah. Other one is ready to prepare, ready to consume, yeah. So all those type of products are made. So in all these processes, there is always a side stream, yeah. That's solid side stream. So either we fractionate it, convert it, or use a fermentation technology to produce fish protein. So how do we obtain fish protein?
You asked this, like one thing can be included, like in Bengal, the cuisine is we use all the bones and heads of the as well as the gills to make a dish with the dal that is called mudo ghanto. Means all those things which is going to be the waste, these are added into the dal and it is consumed.
And then bone is digested inside. Then bones are thrown out.
Later it will be something it may be chewed because it softens while the dal is being prepared.
Correct, all the calcium goes into it and that's there, yeah?
Yeah.
And then still some of the bones are left. When somebody,
Yes,
if you are eating chicken or something, then bones are left,
Yes,
yeah?
sir, yes,
Then the
sir.
boneless chicken and also that's bone. That's
Yes,
again.
yes.
So that means again that side stream comes at domestic level, yeah? Completely agree with all those type of product which will again generate some of the side streams, yeah? So if you see this particular stream, so fish protein extraction, whether you take a whole fish or you take a side stream, yeah? There is a process to produce protein, yeah? We'll cover in detail of this process once you do the experiment, yeah? Once you do experiment, you'll see how simple it is, yeah? It is not complicated. We already discussed how we can fractionate lipids and oils, and we'll go into details when we go for valorization for each and every process. Protein, hydrosylates and derivatives, yeah? So that means how you can develop protein hydrolysate by using specific enzymes, yeah? That is proteolytic enzyme to break the proteins. Then if you have shells, you get chitin and chitosan, or pharmaceutical product in the form of peptides, astaxanthin or certain small bioactives. Now if you look at this particular stuff, lot of research has already been carried out. So remember this, what TRL stands for? Technology, readiness, level, yeah? Number starts from 1 to 9 and this concept has been adopted from NASA. That was the original concept. That means 1 is when you get some new idea, yeah? Not the cellular connection idea, but normal idea. Oh, this will work. Then 9 is deployment at the commercial level. So that is 1 to 9 scale. So majority of the activities are carried out at 3, 4, 5 and 6 level. And this will come in later on when we when we go and look into technology development processes, yeah? So that is TRL level 1 to 9, yeah? Other one, we did discuss about the wastewater which is not being looked into. at the moment, most of the wastewater goes for conventional wastewater treatment. And after this lecture and the next lecture, you will be able to see how we create the value chain and how we can produce microalgae, duckweed, or macroalgae itself on a biomass from the wastewater to generate different chain, and again use the similar techniques to obtain different ingredients. Yeah. So this is the full system, which is a sort of a zero waste approach, which you will try to separate in a cascading manner. The still question is whether we should get protein first, or we should get oil first, or we should get pharmaceutical product first. What should be extracted first? Yeah. That is all the thinking process. You know the technology? Yeah. It is something like, what should be the sequence? When you get up in the morning, what should be the sequence? You have to take a shower, you have to put on the clothes, and then you have to go to your office. So can you take a shower, or can you put on your new clothes first and then take a shower? Orders, yeah? Similar thing, when you're establishing a process, you need to know what should be done first. Yeah? So some of you are doing bachelor's, master's, or a PhD. Yeah? Can we do directly PhD, and then I will when you get married and all, you have kids, you can do the schooling with your kids? Yeah? Not possible, right? So that's why we need to know the sequence and what way to establish a sequence. Yeah? There is no formula for that. There's simple formula: get convinced or convince other person. There's nothing in between. Some cases you can extract oil first. If you do not extract oil first, and if you add an alkali, what will happen? Where is the protein extraction person? If you do not remove oil, and alkali is used for extraction of protein, and if I add alkali directly to the biomass, don't think about the fish, any biomass, the same process, protein is the same. All protein are made up of how many amino acid? 36? 22. Can you name it? Yeah? Good one, yeah.
Tryptophan, tyrosine, phenylalanine, valine, lysine, glycine, and glutamate, aspartate,
Yeah.
asparagine, and
Good, that's
methionine, proline,
Hmm.
and leucine, isoleucine. Just I
Leucine,
remember.
isoleucine, and all those things, right?
Yes.
So when we were studying, we used to make an abbreviation. Met will
Yes.
fly. Yeah?
Yeah. Sir, I just tried animation for remembering.
That's good. So there are almost 22 amino acids are there.
Yes.
And every protein is made up of those 22 amino acids, right? Basically it's 20 plus there are one or two. One is, have you heard of taurine?
Taurine.
Yeah. Taurine. If you see the Red Bull drink and most of those sports drink, yeah, taurine is present there, yeah. Taurine is not a part of any protein. It's a alone sulfated or sulfur containing amino acid, which is mainly found in animal origin products, yeah. And if you isolate it, and this is one of those important ingredient which is used in a lot of different types of drinks, yeah. So that's one of those amino acids. Normally we all know biochemistry says 20 amino acids,
Yes.
yeah. You have essential, you have non-essential amino acids, yeah.
Yes, sir.
So all the proteins are broken down into amino acids, and these amino acids are again used for making protein. What else? They provide energy as well, yeah. If you remember your NCRT textbook or CBSE textbook, there used to be two figures, one Kwashiorkor and Marasmus, yeah.
Yes.
That is when protein is extracted from the body to produce an energy, yeah. How do we obtain energy in human body? Glycolysis. That means you have glucose. What's the next stage?
Krebs cycle.
Correct. Within glucose 1 phosphate, glucose 1,6 phosphate,
Yes. Fructose 6-phosphate, fructose 1,6-phosphate,
Yeah.
and after that,
Phosphoenol pyruvate is the terminal end to make a
is
pyruvate.
it
Yeah.
is a second last step.
Is a pyruvate, right?
Yes.
Pyruvate is converted to acetyl-CoA,
Acetyl-CoA.
and then all this one you have your succinic.
It will
Glutamic?
we go on Krebs cycle, succinate, isocitrate, citrate.
Brilliant.
Yes.
And this one produces NADH, FADH, and ATP.
Yes.
That all goes through electron transport chain.
cycle.
NADH gives 3 ATPs. Yeah.
Sir, it depends 2.5 ATP, and it's just like FADS to 1 ATP, and sir, NADS will be provide 2 ATP, and some cases provide 2.5 ATP. It's depend like sir, we talk about the glucose glycolysis cycle will be produced 32, and in case it will be produced 36. It's depend how much we just like how much we calculate, just like NADPH 2.5 yeah 2, just like ATP.
Brilliant. So that's basically in the broader sense, that is one molecule of glucose will give you around 36 to 38 ATP. This is what is one.
Yes.
Then again, if you go into technicality of it, FADH, NADH, how much ATP it produces. Intention here is when you have a Krebs cycle, some of those amino acids goes directly into the Krebs cycle and starts producing energy. Yeah.
Yes, sir.
That's why we classify amino acid as essential and non-essential. That means it has to be supplemented from external sources. Because of those presence of essential, non-essential amino acid in external source of food, that dictates whether the protein is of good quality or of bad quality or PADCAS value and all those biological value. Am I correct in saying that? That's why egg and milk, they contain essential. That's why we classify it as a better source of protein. Yeah. Because pulses are always consumed with the rice or with the chapati, because pulses they can lack certain amino acid, whereas
Lysine.
exactly lysine or sulfur containing amino acid. So that means our grandfather's father, they were much better nutrition scientists. They knew that pulses should be eaten with the rice. Yeah. Okay. So that's the reason going back. So this will happen in the coming one. We go forward and come back so that you have how we bridge a gap between your fundamental education, what you already have, and how we develop into processes, right? Now, I can give you one question to find an answer. How much 100 grams of food, any food, will give you energy in terms of ATP, not kilocalorie. That we all know of this one. You know, we look at the calorific value. Oh, it contains 10% fat, 10 into 9. Yeah? That is physical form of energy. Yeah? Kilocalorie is a physical form of energy, but the biological form of energy is in a form of ATP. Yeah? So if anybody can calculate 100 grams of a food, how many ATPs it will produce, yeah? Take any food. We'll put it as 100 grams of, 100 grams of fish, yeah? 15% protein, yeah? 80% moisture and 5% fat. To make life simpler, make it fat, yeah? You all know fat goes through lipid oxidation, it produces your ATP. Protein will go into this, yeah? Or even if you change the food product to carbohydrate. I can bet you it will be very, very difficult to get number of ATPs, yeah? Because there will be one fundamental principle which we will forget, and I am pretty confident you all will, well, not all, may forget, yeah? Grams, moles, molecules, yeah? That all will come into play into this, yeah? Let's see, we'll see tomorrow if you get an answer, right? Now back to the business. So that's why when I was asking if we add alkali directly without removing lipid, what will happen? Any idea? Saponification, right? Somebody, because that's the way soap is made. Oil, yeah, fat, you add an alkali, you make a soap. And if saponification happens, you cannot separate a protein, yeah? That's why removal of fat becomes an important entity. If you want to make a fish soap, that's different story, yeah? So all those steps depends what will be the potential implication of that particular unit operation, yeah? So any questions so far with this particular diagram? No? Now, so if we go back to the streams, your fish, shells, leftover biomass, seaweed, algae or bycatches. So these are the broader category of biomass what we get, yeah? So what could be the valorization approach? This is again the same stuff in a different form. There are number of different technologies. So yesterday we discussed briefly about cavitation, that is ultrasonics, yeah, pulse electric field, high pressure processing, what else? Cold plasma, this is here, yeah. Ultrasound is done here. Any other technique? Was the breakfast not very good today? SCCO2, yeah? Supercritical. Or you can have pressurized liquid extraction. There are a lot of different, but the way it sounds looks very complicated, but technically they are very simple techniques, yeah? You work on a simple principle and we get that one separated, yeah? If you have, how many of you use microwave at domestic level? Have you used microwave for heating food? Yeah? Okay. Microwave is one of the most common equipment which is found in almost every kitchen, yeah, to heat a food. But if I put 100 ml of oil in microwave, yeah, and if I put 100 ml of water, yeah, for 1 minute, suppose at 250 watt, just an arbitrary unit, which one will raise the temperature at faster rate? Water. How many of you agree with your friend? Or oil? You agree? Yeah? 3 votes. No, you are not giving vote. That's what it answers. The principle is very, very important, right? Microwave operates at 2 frequencies, yeah? That is 950 and 2450. What is the unit? Megahertz, yeah, right? Because it's 2.4 gigahertz or 2.4 megahertz, 950 megahertz. Why not other frequencies? In processing, only 2 frequencies are allowed. Other frequencies are left over with our Bengali friend to listen to radio FM, yeah? You know, when you tune in the radio, it says this much megahertz and all, right? So other frequencies are used for communication, other purposes, yeah? So what does 950 means? That means polarity is changing 950 into 10 to the power 6 per second, yeah? Right? So if there is a water molecule, what is the structure of water? Very good. This is the easiest structure what we all studied all the time, right? Now, what's the angle? 109. Yeah, 109 or 108 point something, right? but that's immaterial anyway for this particular purpose. That's correct, yeah. Now, when we change the orientation plus minus, it moves very fast. This finger temperature may be around 20 degrees Celsius outside. This is again 20. If we rub it, temperature finger increases, temperature of this increases, yeah. If I rub with the speed of 915 to 10 to the power 6, what will happen? It will heat up very fast, yeah. Now we make a twist to it. If I add 1 gram of salt to it, so it will heat even faster, yeah, because we are adding Na plus and chlorides, yeah. So that is why, and okay, not that is why, but other day somebody suggested that if we give a microwave treatment, we can have a better oil extraction. So that statement is correct or wrong? Don't worry, I am with you anyway, yeah. Loud, 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 yeah. Quick. That is correct, because there we are not heating the oil, we are heating whole of the biomass, yeah, which contains protein, water, salt and all those minerals. They heat up, yeah. When they heat up, it separates the oil out of it, yeah. So the microwave extraction is more towards the thermal side of it rather than microwave on its own, yeah. So I'll go through with these technologies in, I don't know the lecture number, but by coming days we'll have all those technology, so that it does not sound something out of blue, out of moon. That's why I'm just giving some brief touch up with the technology, so that things becomes easier and it won't be a monotonous communication, yeah. So any of those technology, key ingredient we can obtain is protein, oils, fatty acid, chitin and chitosan. I wrote it omega oils because that's the way things are sold in a market. So oil, and difference between oil and fatty acid, you should be very clear again. What is lipid? What is oil? Yeah. What is fat? And what is fatty acids? Yeah. So my eyes are very good with these glasses. So whosoever is skipping, I'm going to ask only them the question moving forward. Yeah. Or whosoever is having very nice jokes on their phones. Yeah. So that's, you remember what is the difference? How we can make a fatty acid out of oil. Yeah. It is not that we can only make it from fish. We can also make the same quality of good fatty acid from your chickpea, almond oil, argan oil, all those ones, or even normal vegetable oil. That means to remove glycerol. Enzyme used for that is lipase, yeah. And what is lyase? Joints, yeah. Lipase, ligases, yeah. Ligases, lipases. Yeah, go, yeah, correct.
Yeah. So just like in DNA replication, DNA ligase just add to nucleotides with the help of NADH, right? No, sorry, NAD. It converts.
That means you need a catalyst to do that,
Yes,
right?
sir.
Yeah. So that is, one is for breaking, one is for adding, yeah. There's a group of enzymes, proteases to break the proteins, yeah, right. So that's the way how you can see how your previous one helps in developing an extraction system. So you need not to be a chemical engineer or a mechanical engineer to understand the technology, yeah. So that's why food science should have all those knowledge, or at least the basic one, so that you can develop your own processes, yeah. Now, solid side stream, shellfish again, we saw the fish one, and then shellfish, head, tail, cephalothorax. Zoologists will say, what is cephalothorax? Something called hexapoda. Yeah, right? When you have head and thorax together, cephalothorax, yeah? Hexapoda means not the Tamil poda. Poda means go, right? Hexa means 6 legs, yeah? So anyway, all those animals, yeah. So we use different technique, that is enzyme assisted extraction, ultrasound assisted, that means the technologies are added, pulse electric field assisted. So here electric field is given in a pulse mode, yeah. HPAE, HP, high pressure processing or high hydrostatic pressure processing. Well, both the same thing, the terminology difference. And supercritical fluid extraction. What is APP? When there is high pressure processing, another one is atmospheric pressure processing. That means done it in natural environment, yeah. So don't get bogged down with these abbreviations. Sometimes scientists, including us, we have a tendency to complicate things, yeah. That's why it says कहना क्या चाहते हो. That means what you want to really say, yeah. Right? So that's one. And all these techniques can be used for protein, lipids, chitin, chitosan, astaxanthin, and other compound. Now, what other, when it mean by other compound, we did cover briefly protein, lipids, chitin, astaxanthin. What other compounds? Yeah. Sorry, Mike. Yeah.
Sir, pigment, just carotenoids.
Carotenoids. Okay. Will astaxanthin not be called as carotenoids?
Sir, it's a type of pigment.
True. That's correct. Yeah. Carotenoids from microalgae as well as macroalgae.
Yes.
Yeah. Any other compound?
Sir, another compound like.
Yeah.
Sir, like lipid, carbohydrate, proteins.
These are done. Other compounds. Yeah. Some are even peptides are there.
Yes.
Other compounds, that means all those minor constituents which can be obtained. Yeah, that's right. You have minerals, you have vitamins.
Yes.
Vitamin is not written. What are vitamins? They are vital amines. Correct? That's the way the name came in. And some of those are small minor carbohydrates are there. What is basically a carbohydrate?
No, it's a monomer.
No, define this word.
It's basically made from carbon, hydrogen, oxygen.
So that means hydrates of carbon
Yes.
becomes carbohydrate. So if you have black coal, yeah, that's carbon.
Yes.
You have water, you add a water, it becomes carbohydrate. Yeah,
Yes.
essentially, right? What that means, if you are overheating a product, what happens? It becomes black. Am I correct in saying that? So basic formula of carbohydrate is CH2O
N whole.
N,
Yes.
right? If it is 6, it becomes glucose, yeah?
Yes.
So only nature is having that power to take the carbon and with the water to make carbohydrates, yeah? So we cannot add water to the carbon to make carbohydrates, yeah? So why I am mentioning this here, when we estimate a moisture, we say suppose it contains 10% moisture, yeah. Why these elements are very, very important in order to establish a biorefinery, to see what is the mass balance. If there is 100 kg of product, so you should be able to get 100 kgs of different substances. In inaugural address, when Professor Panda was mentioning the efficiency is only 30-35%, yeah. Other things are gone as a waste. And you know, first law of thermodynamics, second law of thermodynamics, nothing can be created, nothing can be destroyed. We can only change the forms, yeah. So when we estimate a moisture, this is one of the limitations why a lot of mass balances, they go wrong. Because biomass, if it contains 20% moisture, you get an ingredient which contains 5% moisture, yeah. So when we say moisture content, what does it really means? Is it total water content?
Unbounded means surface पर जो पानी होता है उसी को.
Correct. That is basically the three different types of water there, right? One is free water, bound water, and okay, what free in the water is, that's fine, it's present in the cells, yeah? What is unbound water? So, see, it's good that we know all those terminologies and what is it. But if you have an apple in a hand, if you squeeze it fully, you get your free water, yeah? Then what will be a bound water, what will be unbound water? Yeah? You have to repeat it again, sorry.
Okay.
Yeah.
Bound water is the water attached to the chemical constituents of the food, which can be separated during the drying process or any other processes. But the third category of water, I don't remember the name, that cannot be separated from the component.
It can be separated, but you will be left over with the carbon. This is called bound water. Yeah?
Yes.
The three water, one is free water, and we normally estimate the free water as a moisture estimation. So there is an AOCS method or AOAC method, 105 degrees Celsius for 24 hours, yeah? Then you keep it into a vessel to determine the moisture A, or else 121 degrees Celsius for 1 hour, yeah? That is for grains. For oils is a Carl Fisher titration, yeah? All those who are doing masters, they will be pretty much very clear about this. Proximate composition and ultimate composition, yeah? When it comes to sugars and chocolates, we cannot use such temperature. We use vacuum oven method, because if you use 121 degrees Celsius or if we use 105 degrees Celsius, all the sugars will be broken down and you report it in terms of fat content, or sorry, moisture content, yeah? So the bound water, unbound water is basically water between the fibers attached with van der Waals forces. Any fiber which absorbs the water between the molecules. One protein molecule with lipid, lipid with lipid, or protein lipid. That's all is bound water, sorry, unbound water, free water and bound water, right? When we get rid of bound water, only carbon is left. That's why when we overcook bread or a chapati, it becomes black. That means that bound water is gone. We are only left with the carbon. When we get rid of carbon also, then what is left? Ash. That is basically a mineral, yeah? And that contains all your iron and everything there, yeah? So that's the way how it works. So that all decides how we can get this. And another important minerals what we can get out of this is your selenium, yeah? When you buy those mineral bottles, you know, like it contains good amount of minerals, from where they come in, yeah? All these sources, heavy metals and everything will come into ash, yeah? And then you have your feed additive, fertilizer, pharmaceuticals, packaging, cosmetics and different ingredients. All these ones can have different applications, yeah? So you have 3 or 4 challenges being a food scientist or a food engineer, or whatever you want to call yourself. Biomass, how we process it to get ingredients. Once you have ingredient, what you do with that, yeah? First you need to earn the money. You earn the money. What you do with that money? You bought a car, then you bought a car, what to do with that, yeah? So there is always a challenge. It's not only in this case, yeah? So that's one, and we'll go with the shrimp shells now. Harvesting, storage and frozen. One thing is good, lot of things some of you have already covered, so it makes things much easier for me, right? Shells, harvesting, thawing and washing, peeling, cooking, and goes to market, yeah? This is all is a part of the big project we're talking about, impress-he.eu, yeah? You can go to the website, you can have a look, all those update, and this will running till 2027. and all those commercial processes, how they are being transferred not in the lab alone, at the pilot scale to the commercial, and commercial to the business running. Yeah, this stands for 6.8 million euros, and it's a big money. Yeah, and only focusing on blue circular economy. So there are almost 80 plus researchers across Europe are working on this one. Yeah, these are big projects, something like what you see, big project for establishing, factory or a biorefinery and all those. And based on these outputs of this, whatever value chains are created, then the factories are established. Yeah, you'll see some of the example in coming stuff. So wastewater generated 8.5 liter per kg. Yeah, rough estimations. Yeah, that will vary depending upon your processes. Wastewater 1 liter per kg depending upon the cooking process. Yeah, that means you have shrimp shells, you wash it, clean it, put in a water and you cook it. Yeah, it's something like when you cook a rice. Yeah, if you have 100 grams of rice, and how much water you add in a rice? With this finger? Yeah, I only know this. Whenever my mother used to ask to make a rice, she'll say, put the water till this one. I never understood that unit, but that works always perfectly. Yeah. So that means if you are adding 1:5 or 1:6 ratio or 1:10 ratio. Yeah. You cook it, and this is again optimized so that you don't have amount of wastewater generated. And in that process, shrimp shells 600 grams per kg, 75% of moisture. You dry it, mill it, demineralize, deproteinize, acetylation. And this is what you are going to start doing the experiment today. Yeah. So now next time when we are coming in a lecture, you are going to tell me what does it all means. Yeah. And how do we achieve this. Yeah. Shells, you dry it, and you all are lucky that some of researchers from NIT, they are not here at the moment, they have already dried those shells for you. Yeah. And then you grind it, you demineralize it. That means you use a technique to remove the minerals from there. The shells are hard like a stone, but you remove the minerals. Then what will be left is a protein and chitin. Then you separate the protein. Then you are left over with the chitin. And that chitin is converted into chitosan, which is sold in a market. Yeah. So shells are waste. They just throw it in a bin. And from the same shell, you make a chitosan. What is the cost of chitosan? Any idea? When you are doing experiment, ask those questions. Yeah. They all know those. Yeah. And this chitosan is also used to make biodegradable packaging material. They are also antimicrobial. They use in agriculture application as a fertilizer as well as biopesticide or biofertilizer. Yeah. And these extractions can also be done by ultrasound, microwave, combination, fermentation and enzymes. So what you will be doing is only the basic part of it. Then it is left on to you how and where you can use those technology. This will be covering in theory. Yeah. And each and every topic will have a PhD topic on its own. Like your friend is working on thermosonication of milk. Yeah. So that again itself is a full thesis topic. Yeah. Now and then you have your chitin. So if you start with a kg, you can get up to 45 grams per kg of wet shells. Yeah. So these are all goes with the economics and economic assessment. So what are the side stream we get? Protein rich water, mineral rich water, yeah, and alkaline water. Yeah. And this will give you mineral. What is a calcium? Source of calcium. Yeah. And then this will give you the protein, and this shell protein is having strong bioactivity. If it is converted to peptide, it's having a lot of bone healing properties. Yeah. And then alkaline system. So this is what we can get. all those ingredients. And you mentioned about the pigments, carotenoids, and you get it from shrimp, that yellowish color pigment, which is again having a biological activity. Yeah. And then this is some of the calculation. You don't have to worry about the numbers. 5.6 tons of shrimp shells generated. This is for a specific country. I won't name it here now, yeah, because certain informations are a bit confidential as well. And then this much cubic meter. So, who's good engineer here? 1 cubic meter means how many liters? Or 1 liter is equal to how many centimeter cube? That should be like this, 5 tens are 50, like that, yeah. So 1 ml is equal to 1 centimeter cube, right? That means 1 centimeter by 1 centimeter cube, you can put only 1 ml of water, provided the density is 1, yeah, 1 kg per liter. Oil, that is volume and density. You don't agree? Yeah. So if you see 5.6 ton, that is 53.2 metric cube of water is used. This is the amount of the wastewater which is generated. Yeah. This many tons of shrimp shells, this many kgs of your 840,000 tons of dried shells, and how much chitosan you can produce, yeah. And if 1 kg of shell of chitosan costs around 5,000 rupees or 6,000, I know in terms of euros, crude chitosan we get around 100 euros for a kg or something. So you can think of how much is the value which is going into the bins, yeah. And this is not a rocket science to produce a chitosan. It's been done since for last 50-60 years, right? So only to take it and make a business case, yeah. Total side streams generated, yeah. So this is one of the example of shells, and we'll take a break. And what we have covered so far is all your side streams, and given a specific case of shrimp shells, how we can get into chitin, chitosan. And when we come back, we'll go through again with liquid and bit of solid together, yeah, because both are produced hand in hand. So that means if you are extracting chitosan, there is again a water which is produced. How we can valorize it, yeah. It is something like heat can be extracted. You use electricity to light bulb. Bulb produces a heat. Can we extract that heat? Yeah. So that's like again, where do we stop it? Yeah.